Okay, welcome to module 1.2.1. This is going to be plant metabolism and photosynthesis and how we absorb sunlight so that we can produce glucose and what are the processes towards that. One of the big things that I've kind of come to a realization, someone pointed out to me that the whole purpose, like our goal, is to harvest as much sunlight as possible and efficiently convert that into energy. I feel like sometimes when we think about crop production, we're thinking about we need to fertilize and we need right soil, and we can have all of those things, but without sunlight, that plant does not perform photosynthesis. So by now, we should be familiar with the photosynthesis equation. We need 6 carbon dioxides combined with 6 water molecules. And that carbon dioxide plus water, through a series of events, will be converted into sugar or glucose, C6H12O6, and one of the byproducts of that will be oxygen. So that is kind of one of the premises of wanting to increase our plant populations so that we can minimize or reduce or decrease the carbon dioxide in the atmosphere. And so by that, we give off oxygen, which is what humans need to breathe. So there's kind of this back and forth. We give off carbon dioxide, plus human activities increase the carbon dioxide concentration in the atmosphere, which is actually less than 0.05%. So we have carbon dioxide, we have water from the soil going up the xylem, and inside of that leaf, we need chlorophyll. One of the most important portions of this is chlorophyll, because if we were to take carbon dioxide and add water or carbonated water and just put it out in the sun, we wouldn't get a glucose molecule. We need chlorophyll in order to create this glucose molecule. So the thought processes are, what are the inputs? What are the outputs? So this is in, we need chlorophyll, and the outputs are going to be sugar, and finally oxygen is expelled through the stoma and added back to the atmosphere. Many of us are familiar, at least with this from plant science. Again, a simple diagram is carbon dioxide in, we have water from the soil and nutrients. Light energy is then converted into a sugar, which can be used and stored in the rest of the plant, which is also where we get our energy from. Us as humans and as animals get their energy from, and we give oxygen back off into the atmosphere. So this occurs in the chloroplast, and we'll kind of go through a couple of the light reactions and the dark reactions, and just a brief overview of what happens, where it happens, how it happens, to finally get to that glucose molecule. So as I mentioned before, we have the light reactions, or also known as the LDR, meaning light dependent reactions. These reactions require light and only happen during the daytime, because this is the first part of the photosynthesis where the leaf is adsorbing that light and then doing something with that. So we have the electron transport in the Z scheme, and that might be a little bit too much for us in this particular module, but just to understand that there are two separate independent reactions that have their own specific requirements. So the light reactions must have light. The primary electron transfer, we have some energy stabilization, and that's really getting into the cellular level. If you watch the video that I mentioned that you had to watch in order to get to this, you'll see where all those photons were actually striking and going into the plant, and then it was moving along the Z scheme. And then ultimately, that transfer of energy and that movement in order to get the energy into the cells in the chloroplast. Next, we have the dark reactions, and these are LIR. They are light independent reactions. So these can happen during the daytime or during the nighttime. So we have the LDR, the light reactions, only happening during the daytime, but it's not like that's the only thing that's happening. So we have energy coming in from the sun. We have the plant absorbing all of that light, at least the red, the blue, and they are, I mean, there's so much energy that's going on in that like it almost can't keep up with, the dark reactions cannot keep up with the amount of energy that's being provided. So once that light is received and absorbed with a B, I'm sorry, absorbed with a B, the light reactions start to happen, and then the dark reactions in the Calvin cycle can happen right behind that. So while the plant is growing during the daytime, it is also getting some of the outputs, that glucose of the Calvin cycle, while it's actively absorbing that sunlight. Then, once all of that sunlight is absorbed, it still has to work, well, what might be considered overtime in order to keep up with all of the energy, or to process and metabolize all of that energy. So we have the light dependent reactions. They occur in the grana and the thylakoid. Light dependent reactions happen in the grana and the thylakoid. And they produce NADPH and ATP. These are what drive the Calvin cycle. You see in this, we have ATP, that is energy going into the Calvin cycle. We have this NADPH and this NADP that's kind of spinning back and forth. And you got to think about like, I guess maybe a cog or a wheel, that as this moves in, something happens. NADPH goes back into the grana and the thylakoid, produces NADPH, which helps spin the Calvin cycle as a part of that. And then we also have some other metabolites that are being produced that go back into the grana and thylakoid as well. There are two phases of this. There are two phases of the light-dependent reactions. One is going to be phytolysis of water. The next will be phosphorylation. All right. Not that you really need to be able to go into like explain what photolysis or photolysis of water is, but water is split into hydrogen and oxygen, and the byproduct that we're looking for is this electron worth of energy. So when that oxygen diffuses out of the chloroplast as the byproduct, what is left is energy and this hydrogen. And as part of that photosynthesis of the glucose equation, C6H12O6. That hydrogen will also be part of this NADPH, which creates that chemical energy from previous. The NADPH2 or NADPH is the energy or the driving force of that. This is going to be used as a driving force, a key component of the Calvin cycle, which we'll talk about in the dark reactions. And then we have the phosphorylation, and this is where that light energy is converted into chemical energy and creates this ATP. So the photolysis of water is important for the NADPH. The phosphorylation is important for ATP, or adenosine triphosphate. Phosphorylation, adenosine triphosphate. All right, next, we're moving on to the dark reactions. And remember, these happen all the time. These are not dependent, they're independent. And that occurs in the stroma. And so the stroma being not really a part of a structure, but kind of in um, like in solution inside of this chloroplast, right? It's just a matter that it kind of floats around and or happens in the kind of like the liquid portion of that chloroplast. So that chemical energy, this NADPH and that ATP, this is from the photolysis of water. This is from the phosphorylation. And they both power the Calvin cycle, which ultimately creates glucose, C6H12O6. Okay, moving into the Calvin cycle. And so there are several steps, and there are even more steps that go along with this. But for the most part, we need to understand just a couple of things. Number one, that carbon needs to be fixed. So when we think about C3 and C4 plants, that C actually stands for carbon, because that is going to be the first molecule that is formed. Is it going to form a 3-carbon molecule or a 4-carbon molecule? So the first step is going to be carbon fixation, and that carbon dioxide is going to be bound to RUBP with the help of an enzyme called Rubisco. Not need to worry about that right now. We'll get into it as I break down each one of these phases. Next, we have the reduction phase. And so whether it's a 3-carbon or a 4-carbon molecule, eventually we come up with either 1 or 2 3-PGA. Next, once we have our carbon fixation, we form 2 3-carbon or 3-PGA molecules, and that 3-PGA is converted into G3P, and here comes the energy, and here comes the NADPH and the ATP from the light reactions. Next, that G3P will be turned into the carbohydrate. So this is one of the most important molecules or compounds that come out of this Calvin cycle. And so when we think about carbohydrate formation, naturally we're going to think about glucose. But we can take that glucose and bind it with other sugars in order to form fructose or sucrose and other carbohydrate type compounds. But this G3P is what's going to be important. Here we have more energy from the light reactions that kind of spurns that again, and we're going to continue making more RUBP. So this is this G3P molecule that forms that carbohydrate. There are going to be G3P molecules that continue on throughout the process. G3P plus ATP gets us back to the beginning of the Calvin cycle again. And we're going to go through what each one of these means in the upcoming slides. All right, so a look at it again. We have the light reactions. Plant absorbs light, goes through photosystem 2, the electron transport in photosystem 1, gives ATP and NADPH in order to continue that cycle. But remember, there's also going to be ATP and NADPH that is floating around in this stroma because the light reactions are occurring. So we're producing a lot more ATP and NADPH than the Calvin cycle really needs for one turn. Yeah, so photosynthesis was this really cool thing where plants turn carbon dioxide and water into energy, and it's all fun and the picture's all pretty. But when we break it down and we really start to get into it, there's a lot more to it. So I'm trying to give somewhere in between, it's just C6 or carbon dioxide and water that yields glucose and oxygen. So inside of the leaf is going to be responsible for the absorption of that sunlight. And so if you have a very thick material, that sunlight might not be absorbed as efficiently. Next, we have the chloroplast. Not that you need to really know, like these aren't going to be on the test where we're like, this is the outer membrane or this is the granum. Just know that the thylakoid stack contains the granum. Each one of these little discs is a granum within the thylakoid stack. So like imagine having a stack of quarters or a stack of pancakes inside of the chloroplast. And again, the stroma is just the material that kind of gives it its structure and its shape. All right, so next, the way that we classify these plants by either C3, C4, or CAM. As I mentioned before, we have a C3 molecule. That is going to be the first molecule that is made in step 1 and 2, or a 4-carbon molecule, and we'll see this later on. And then CAM plant is really time regulated. So in C3 plants, we have unregulated. Nothing is stopping that. Carbon dioxide is coming in. As soon as it comes in, it goes straight into the Calvin cycle. For C4 plants, we have a concentration regulated, and that carbon dioxide is going to be stored in the mesophyll cells until the Calvin cycle is ready for it. And then finally, CAM. And CAM is a time-regulated plant. So CAM plants are like pineapples. They are also like cacti. I said that right, cacti. And they are going to take in carbon dioxide at night, make that C4. Then that carbon dioxide will be introduced into the Calvin cycle during the day. So CAM plants and C3 plants are kind of opposite of each other, because where CAM plants grow, it's very hot during the day. Then, as we mentioned before, as temperature increases, as humidity increases or decreases, that's going to influence transpiration. So if cacti were to open their pores during the day, they would lose everything. The water would evaporate so very fast. That's why they have that thick cuticle, that waxy skin, to kind of give them an extra insulation against losing all of that oxygen. And so this is why CAM plants perform much better during a drought, is because they can hold on to their water and their carbon dioxide, and they can perform photosynthesis as they need to. So they can really regulate these processes within themselves, and they're not as susceptible to the environment. So, thinking about CAM plants as we move into warmer temperatures with less water, that pathway is going to be at a greater advantage. Okay, let's look at the C3 pathway for carbon fixation. And I'm doing this because I'm putting this in your brains that the first step is going to be carbon fixation. And so just kind of flow with me here, just to kind of have an understanding that we are going to make a 6-carbon molecule. So this carbon dioxide will bind with RUBP, which is a 5-carbon molecule. 5 plus 1 equals 6. This oxygen will be expelled. And then that 6-carbon splits into 2 3-carbon molecules, or 3 PGA. 5 plus 1 makes 6. 6 divided by 2 equals 3. We have Rubisco. And then that carbon dioxide is going to be fixed as part of a carbohydrate. Moving into phase 2, the reduction phase, this is where ATP and NADPH reduce 3-PGA to G3P. NADPH and ATP from the light reactions are going to reduce 3-PGA to G3P. G3P will continue on into step 3. You do not need to know the chemical name for that, just that G3P is going to form that that carbohydrate. So this is where this is important for animal nutrition, and whether you are in animal science or whether you are in crop science, what we don't recognize or realize is that for the most part, the grains that we grow, though most of the corn and the soybean that we grow, go to feeding animals. We do not consume that much corn. Just think about it. How much corn do you actually eat? Not a whole lot. But some of the products that you do eat are made from corn because they contain these carbohydrates. So we have fatty acids and glycerols. We have a simple sugar as glucose. We can make fructose, and then glucose plus fructose equals sucrose. This is some biochemistry stuff. Not necessarily that you need to understand or know what toast you're making. For the most part, we know we're going to make glucose. Next, we have starch and cellulose and other amino acids. And if you remember from soils, remember this is all of the decomposition. These are all the components of the plant, how they break down. So we got our sugars, our glucose, which ultimately make some of these other compounds as they begin to metabolize or they begin to get synthesized into different structures of that plant. Okay, next, RUBP must be replaced. So from that, we're going to take ATP with G3P. So remember over here, we reduced this 3-PGA. Now we need to kind of oxidize it so that we can get back to this 5-carbon molecule. So just know that for every 3 turns of the Calvin cycle, we are going to use 5G3P. For every 3 turns of the Calvin cycle, how many G3P molecules will be used? So in order to remake 3 of these RUBP, because remember, we're doing 3 turns, we need 3 more RUBP. We're going to require that ATP. So ATP and G3P are the regeneration phase of RUBP. Okay, one of the major issues with C3 plants is that they suffer or they are susceptible to photorespiration. Remember that the Calvin cycle occurs whenever. It can happen during the daytime, it can happen during the nighttime. It is an unregulated process. So during hot, dry climates during the day, the stoma will close, which means that as that plant is still performing the light reactions, one of the byproducts of that light reaction is oxygen. Well, that oxygen stays in the plant and the Calvin cycle is still going. It will take whatever is in the plant. So if there's oxygen instead of carbon dioxide, there is no carbon to take up or to move into that Calvin cycle. So we consume the ATP, but we don't make the ATP, nor do we make sugar. So if we don't make sugar, that Calvin cycle is basically spinning or, performing that process inefficiently. During this photorespiration, RUBP will fix oxygen instead of carbon dioxide, which will eventually decrease that PS, the photosynthesis output of ATP and sugar. So RUBP is going to fix oxygen instead of carbon. Taking a look at this is going to be in the stroma. We will have carbon dioxide that comes into photosynthesis as part of the Calvin cycle. Carbon dioxide into the leaf. Rubisco fixes it, makes two 3-PGA molecules, which eventually get reduced to G3P to make a sugar. Carbohydrate formation. Next, we move into the regeneration phase. So we need G3P. I know it doesn't show it here. G3P plus ATP to make RUBP. Well, look what happens if we take oxygen and it gets fixed by Rubisco and add it into that cycle. Notice that the output is a carbon dioxide rather than a sugar right here. These two, this is the bit. So we didn't make our 3-PGA. The output, or in step 3, we produce a carbon dioxide rather than a sugar. And we don't really make back any RUBP. So photorespiration is a very inefficient process. Photosynthesis is efficient because ain't it sustainable? So we take the carbon dioxide in and we continue to make the energy both for the plant and for the Calvin cycle to continue its process. So one of the things that researchers have done is go, oh, well, if we add more carbon dioxide into the atmosphere, then, and this is in greenhouses, if C3 plants are going to suffer from photorespiration, well, we'll increase the carbon dioxide concentration such that when that leaf is open, it will take in more carbon dioxide. So if the carbon dioxide concentration is high, more carbon dioxide would go in through the stoma when the plant was open. When will go into the plant when the stoma is open. And now the stoma here will open at night. For C4 plants, and you notice that they're set up just a little bit differently. Kind of the arrangement of the vascular bundles and the mesophyll cells are slightly different, and we'll see this and why it's important in the next slide. All right, so C4 pathway has the advantage of high light intensity and high temperatures and low moisture, which is going to be what we expect the future the future climate or environment or weather to be like. So the C4 pathway occurs in the mesophyll cells. We have this PEP-A carboxylase. This PEP-A carboxylase is very important in C4 plants. Because this PEP4 carboxylase is going to hold on to, or it basically acts very similar to Rubisco before we get to the Calvin cycle. So this PEP-A carboxylase will fix carbon dioxide and then transport or move that into and have it ready for when the Calvin cycle needs it. So that PEP-A carboxylase is going to fix the carbon dioxide to PEP, which is going to be a a 3-carbon compound, C3, 3C, they're the same thing. So we have a 3-carbon molecule that is PEP. The PEP-A carboxylase combines the carbon dioxide and the PEP into oxaloacetate. And that oxaloacetate will then be moved into the bundle sheath cells, be broken down or being pulled apart so that the carbon dioxide can go back into Rubisco. If we go from oxaloacetate and 1 carbon dioxide comes back off, that C3 compound, a PEP, gets moved back into the mesophyll cells. So this is really just like a transport / drop-off mechanism, this C4 cycle right here that occurs in the mesophyll cells. Fixes carbon dioxide to make a C4 compound. C4 compound comes by and kicks that carbon dioxide off into the bundle sheath cells and then just continues the cycle. So PEP, oxaloacetate, lose a carbon dioxide, PEP, oxaloacetate, carbon dioxide, PEP, oxaloacetate, carbon dioxide. So the Calvin cycle actually occurs in the bundle sheath cells. That's why it's kind of a location dependent, or it's regulated based on the location. So these cells can still remain open, the stoma can still remain open and take in this carbon dioxide. And then it will give it to that Calvin cycle as needed. So this is the storage place for carbon dioxide. When the Calvin cycle is ready for it, that carbon dioxide gets pushed into the bundle sheath cells where the Calvin cycle, the C3 pathway, the Calvin cycle is technically the C3 pathway. This is the Calvin cycle. It is also the C3 pathway. It just occurs in a different spot or location. It occurs in the bundle sheath cells, where in C3, it happens here. That Calvin cycle occurs in the bundle sheath cells. Okay, a couple of different looks at this. We have carbon dioxide coming in through the stoma. PEP carboxylase. We have our 3-carbon PEP or PEP. That PEP carboxylase fixes this CO2, kind of tag teams with PEP to form oxaloacetate. Into malate, the malate gives off the carbon dioxide, and pyruvate, or this 3C, goes back into the mesophyll cells. This carbon dioxide will then perform, or will then be a part of the Calvin cycle. This is going back to step 1, which is carbon fixation, Rubisco, and RUBP. Rubisco fixes the carbon dioxide, attaches it to RUBP, forms a 6-carbon molecule that then gets split down into what? What is the intermediate step between the carbon fixation and the reduction phase in the C3 cycle? Remember, we need to form 2 of these. We have a 6-carbon molecule that then gets broken down into 2 3-PGA molecules. 3-PGA is then reduced to G3P by what two intermediary, or what 2 molecules do we need from the light reactions in order to make this happen? Right, we need ATP and NADPH. That is going to form our G3P molecule that will then be converted into glucose. Next, we have our ATP that is going to regenerate the RUBP binding with G3P to continue this cycle around. So you can see if we didn't have oxygen, I mean, if we didn't have carbon here and it just fixed oxygen into that cycle, none of the, well, first off, we wouldn't make carbohydrates as efficiently. All right. Next we have CAM. So during the nighttime, the stomata will open because the temperature is decreased. There might be some more humidity in the air because the temperature has cooled down some. And we are going, that plant is going to fix carbon dioxide much in the same way that it does in the C4 pathway. C3. We're going to fix that carbon dioxide to form a 4C carbon molecule. We still need the NADPH and the ATP. Sorry. During the night, we're going to make that as a 4C. We're going to fix that. We're going to fix as much carbon dioxide as we can. We're going to kind of just hoard as much as possible. During the daytime is when we will actually perform the dark reactions. So the stoma will be closed. We'll have plenty of water to perform this photosynthesis. We've got photolysis and phosphorylation that make NADPH and ATP. That 4-carbon molecule will release that carbon dioxide. This 4-carbon molecule being something like oxaloacetate from the C4 pathway. And will then give that carbon dioxide into the Calvin cycle, which is the C3 pathway. So this is just a time fixation of the C3 pathway. All right, ending it out with a comparison of the pathways. Remember, C3 is going to be the Calvin cycle. This is the base. This is the original. We have Rubisco as the carbon dioxide fixing enzyme. The Calvin cycle occurs in the mesophyll cells. So carbon dioxide is going to be fixed in the mesophyll cells in all, at least the C3 and the CAM. And C4 as well. The biggest difference between C4 and C3 is the carbon dioxide fixing enzyme. In C3, it's Rubisco. In C4, it's PEP carboxylase, PEP A carboxylase. Remember that carbon dioxide is fixed in the mesophyll and then shuttled into the bundle sheath cells. This is where the Calvin cycle occurs. So mesophyll, remember, and then that's going to form a 4-carbon. That carbon dioxide will then be released into the bundle sheath cells. That PEP will move back into the mesophyll and continue to be fixed with that carbon dioxide via the PEP A carboxylase. And again, the C4 or CAM is like a combination of both, but has been flipped by time. Next, what do we think about photorespiration as far as C3? We think this is going to be high or low? High. We will not have photorespiration in CAM or C4 because remember, we're like stockpiling that carbon dioxide in the mesophyll cell. We're stockpiling it so that the Calvin cycle has enough carbon dioxide in order to efficiently complete that process. So what do you think would be the ideal environment for C3 pathways? Hot or cool? Okay, cool. And so what do you think do we need a lot of water? We would need a lot of water in order to perform this process. Do you think that the C3 would perform better in high carbon dioxide environments or low carbon dioxide environments? High carbon dioxide environments, because while that stoma is open, it can actually take in more carbon dioxide. So cool, moist, and high carbon dioxide environments are going to be most ideal for C3 plants. And so when we're talking about the energy efficiency, because the potential for photorespiration can occur, it is the least energy efficient. C4 plants, on the other side of that, remember, can handle high light intensity and hot, dry conditions. This is likely to be what we're expecting to see in the future. And it is moderately efficient. It is better than C3. And CAM plants perform the best in very hot and dry environments. And it is the best at water conservation. So, these two might go back and forth as to which one's the most efficient, but this one has the advantage that it can hold on to water longer. Because remember, the stoma closed during the day. The stoma stay open during the day, or they are open during the day in order to perform transpiration. And then water loss is high, moderate or medium, and low. Finally, some examples of C3 plants are going to be rice, wheat, peanuts, cotton, and soybean. Well, we sure do eat a lot of these, right? And so what's going to happen, because remember, we said that this is likely to be the environment in the future. What is going to happen to these crops as these environmental conditions begin to be more prevalent? And remember, we've talked about this in class, that we're trying to reduce the carbon dioxide concentration in the atmosphere. But for C3 plants, in order for them to thrive, they are going to need at least high carbon dioxide concentrations. Man, how are we supposed to do this? Going to be tough. All right, next we have our C4 plants: corn, sorghum, sugarcane, millet, most of our grass plants, and then our CAM plants. Examples of that are going to be cacti, agave, and pineapple. All right, so this table right here is just to kind of give you an idea of the differences between the two. A test question might seem something like compare and contrast the difference between C3 and C4 plants. And some answer would be like, in C3 plants, carbon dioxide is fixed by rubisco, whereas in C4 plants, Pepe carboxylase fixes the carbon dioxide. Under what environments would you expect the C3 plants to thrive in? Well, not very hot and dry. Cool, moist, with high carbon dioxide concentrations. List an example of a C3, C4, and CAM plant. Like, I feel like this particular portion of the module is going to be the portion where you're going to have to explain that to me. You'll have to walk through each one of those processes described in this, and those are likely to be short answer, well, medium, moderately long answer questions. This particular module, this 1.2.1, is where I'm going to get the essay questions from. And you're going to need to explain all the way through everything. All right. I feel like that ends module 1.2.1. I would revisit this one several times over the next week and make sure that you have a good understanding of it. Also, when I come back from Atlanta, I'm going to walk through this again and maybe give some demonstrations of that, like in class. I hate that I'm going to be gone for this because this is one of my favorite portions of that, so that I can explain the difference and maybe give some, I don't know, like some hands-on activities as to what happens when that carbon dioxide comes in and what happens to it. Okay, that's it for module 1.2.1.